Dear ladies and gentlemen, I'm a bioengineer and a PhD student from the University of Veterinary Medicine Budapest, Hungary, and the main goal of my PhD was to map the prevalence of a newly discovered virus, atypical porcine pestivirus, among the Hungarian herds, and to compare the domestic and the foreign strains through phylogenetic analysis. We found that this virus is highly distributed among Hungarian herds, and the detected strains are of high variability, causing congenital tremor. So we plan to find a correlation between the virus and the tremors through various histomorphological methods. At first, we try to identify the morphology of the virus through transmission electron microscopy, but we failed to 100% sure to be described this virus. There were other filaments that looked like viruses, but it was not sure, so we made other plans. That's what I'm going to talk about today. So, congenital tremor is a common phenomenon among newborn piglets. It's characterized by shaking of the head and the limbs, and it's usually the piglets are clinically healthy through waning age. Though it's not the direct cause of death of piglets, but it can hinder the piglets to find the teeth to suckle, so it can indirectly lead to growth malformation, growth retardation, and death by starvation. CT is classified into two types, type A and type B. Type B is type A is where there are visible histological lesions in the brain and spinal cord, and type B where there are no apparent lesions. Type A can be classified into other 5 subgroups. Type A3 and 4 is caused by a genetic disorder. Type 5 by a chemical substance, and type 1 and 2 is caused by viral infection. That's where pestiviruses come in. Pestiviruses are high variable aeronaviruses of the Flaviviridae family, which are important pathogens of cloven hoofed animals. Not so long ago, it consisted of 4 recognized species: bovine viral diarrhea virus 1 and 2, border disease virus, and classical swine fever virus. The latter is responsible for A1 type congenital tremor. In the last 2 decades, other atypical pestiviruses has been described, like the bungavanna virus, which was only identified in Australia, the Lilla virus, and the atypical porcine pestivirus, which was first detected in 2015 in the US by next-generation sequencing, and since then, so far, in a lot of countries of Europe, Asia, and America. It was happened to to be responsible for A2 type congenital tremor, and on the affected farms, it can cause up to 30% of piglet death. Here on this video, you can see the animals we got in this summer. The 2 animals marked by red paint are CT affected, and this one is a healthy one. Usual characteristics of CT, that when the animal is asleep or resting, the tremors did not occur, so they are not shaking. Study showed that APPV is mostly localized in the inner granule cell layer of the cerebellum, but not in the outer granule cell layer. It is pretty important, in fact, because in the case of classical swine fever virus, which has really high mortality, the outer granule cell layer is also infected. They theorized that maybe this could be the cause that APPV-affected animals could be clinically healthy through weaning age, because healthy neurons can compensate the loss of inner granule neurons. The aim of our research was to characterize the morphological changes in various brain segments of CT-affected, but naturally APPV-infected piglets by the use of various histomorphological techniques. First, we got one CT-affected and PCR-positive animal from farm from 2 years ago that was 3 days old when we euthanized it. And this year, we had 6 animals from 2 different farms. 2 animals were affected by CT and PCR positive, and there were one and not affected animal, and they were 1 day old. We used them for histopathological RNA in situ hybridization and immunohistochemistry methods, and the other parts of the samples were used for electron microscopic examination and for PCR. We performed hematoxin, auxin, and luxophase blue staining of the samples from the central nervous system, but we found no remarkable differences between healthy and CT-affected piglets. Then we performed GFAP immunohistochemistry on these samples, which signals the oligodendrocytes responsible for myelin formation, like astrocytes and ependymal cells. And then we performed RNA-scope, which is a really sensitive and specific RNA in situ hybridization method due to its special double Z probe design. And after that, we scanned all of these slides with the panoramic slide scanner to have an overall view of all of our slides. In the central nervous system, oligodendrocytes are responsible for the myelination of the axons. They myelinate the axons in concentric layers, interacting with themselves and the neuron along the length of the axon. And if there's some deviation from the normal myelin sheath, then there's a misfunction of neurological function. We use a newly described quantitative method to describe the degradation of the myelin sheath, the G ratio, which is A stands for the diameter of the axon, and it's divided by B. That's the outer layer, myelin sheath in diameter. Okay, so there's our RNA-scope results. It's strong signaling. Can be seen in the inner granule cell layer, in the prokinase cells, and the molecular layer of the cerebellum, both in the 3 days old animal and in the 1 day old animal. And they were all affected by APPV. And none of the negative animals were positive by RNA-scope. So, well, surprisingly, the 3 day old animal cortex was positive for APPV, but the 1 day old animal's cortex was negative, and it was the same as well in the case of the brainstem. We couldn't find out what was the cause of it, but maybe the infection occurred in different times of gestations, or maybe the congenital infection only affects the cerebellum at first, and through ontogeny, the virus spreads through the central nervous system, or maybe it was different strains that affected these animals. We could only know this with further research. Here you can see the GFAP staining of a healthy animal's cerebellum, and then the CT-affected animal's cerebellum. Here you can see the Bergman filaments that are responsible for the mediation of the neurons from the outer granule cell layer into the inner granule cell layer. So it seems that somehow the APPV infection affected the glial cells. Maybe there are fewer glial cells, or their function is lessened. Here you can see the transmission electron microscopic results. It's a lower magnification, but on the left there's seen the negative animal, and in the right the CT-affected animals, and it's clearly seeable that there are degraded myelination in the case of CT-affected cerebellum. Here's a higher magnification. It's the samples of the negative animals. There's concentrical normal myelinization, and here is there's a wavy degenerated structure of myelin sheath. We checked 16 view slides of this in each cases, and we measured over 1,000 axons through the process. This is an example for the healthy animal, and this one is for the sick animals. And there are relevant numbers of cases where the axons were also degraded in the case of CT-affected piglets. So overall, we calculated the G ratio, and we found that in the case of sick animals, the G ratio was significantly lower than in the case of healthy animals. And maybe it's because of the remarkable myelin degradation due to CT. So all in all, congenital APP infection primarily affects the inner granule cell layer and the molecular layer of the cerebellum. And although positivity was found, it was inconsistent in the case of the cerebrum, and that needs further research to be sure that what is causing this. According to our immunohistochemistry and quantitative electron microscopic results, significant myelin degeneration could have been seen in the cerebellum of CT-affected animals, and maybe due to this loss of myelin, the degeneration of the central nervous system occurs, and that could be the possible cause of the tremors. In the future, we plan to perform a complex immunohistochemical and transmission electron microscopy examination of the cerebellum to gain information on the morphology of the virus and the ultrastructure of the involved cells. I would like to say thank you for the travel fellowship that made it possible for me to come here and that my work was chosen as a presentation. I would like to say thank you to Gyula Balka, my boss, and my lab mates Renata Pop, Kitty Schönhardt, and Carlotta Valetti. Thank you for your attention.